Hello, and welcome to our third lecture in microbial genetics, where we get to discuss RNA, the structure and function of RNA. RNA is pretty versatile. Like I said in the former video, it works as the messenger, but it also has some structural roles and some active roles to play as well. So first things first, when we discuss RNA versus DNA, we're almost talking about the same stuff, but we're not quite, okay? So if you look up here in the figure and over there, you can see that we have a deoxyribose, like in our DNA, and it is named rather cleverly, DNA, deoxyribose. Deoxy, no oxygen. And our RNA has ribose, just regular ribose. And it has that OH group there. Okay? And so it contains ribose instead of deoxyribose. DNA, RNA. That's where you get those letters from. Okay? And then right above me here, we can see that RNA does not contain thymine. It instead contains uracil. Okay? So what does that mean? It means if I'm trying to base pair in DNA, I use A to T, adenine to thymine. But when I'm using RNA and I'm getting my RNA apparatus in place and I'm putting it on the DNA to actually do something with that DNA, I'm going to copy it using uracil. Okay? And so that is the two fundamental differences between DNA and RNA. The ribose versus deoxyribose and uracil instead of thymine. All right, going a little more in depth, okay, and kind of diving head on. Okay, DNA is typically double-stranded. RNA is typically single-stranded, but it can form hydrogen bonds with itself and kind of form temporarily these hairpin structures, we call them, with complementary base pairs. But they weren't designed to line up like in DNA necessarily. Sometimes they are, but sometimes you design them yourself in the lab and they are complementary and they ruin your experiment. So you have to watch out for those. They have software to detect them. Anyway, the RNA folding in on itself is because it is single-stranded and those hydrogen bonds beg to be formed. Okay, so single-stranded versus double-stranded is another difference between DNA and RNA. And to use RNA, we need to ask ourselves, what are we making with the RNA? Okay, right? We need to make protein, right? So we're going to have protein synthesis with RNA. And what do we do that with? Our ribosome. So that is why we needed to learn our ribosome back in the cell chapter. And this is how we actually grow our amino acid chain or our peptide chain. And it is because the ribosome makes a peptide bond. And remember I said that we have a messenger RNA. The RNA forms as a messenger. Well, that's what the M in this stands for, the mRNA. It is the messenger from the DNA template that is then going to make the protein. And the protein gets made by reading the codon, we'll see later. It reads what's inside of a ribosome, and it uses a RNA as a structural and active unit in order to grow the peptide chain. And so the ribosome enzymatically puts these close together. The tRNA holding an amino acid and the growing peptide chain, they enzymatically get close together, and with the correct energy and the correct tRNA on the correct sequence, the enzymatic activity will grow the chain with the peptide bond. Okay? And to show you what a tRNA looks like holding a amino acid is this nice handy dandy picture. Okay? It is pairing with our mRNA, and it is reading the sequence there, GAG. And so what reads that GAG but a CUC tRNA? It matches up, just like we talked about. C to G, A to U in RNA, right? Instead of A to T in DNA. Remember, we're going to use uracil instead of thymine, right? And then C to G again. So that'll pair up. The glutamic acid will then grow and pass on with the peptide bond to the growing peptide chain. Or eventually that will be folded into a protein. And that is why we learned about protein. So to regroup before I summarize, we learned about protein, we learned about nucleic acid, and we learned about the cell. And this is why we learned about it all, because it's coming together here. And in the RNA here, we're going to be making protein. And with protein, we can do work and activity. We can evade white blood cells if we're a pathogen. We can ferment if we run out of oxygen. Lots of things happen when we have proteins to respond to certain things. To summarize, RNA has a structure and function depending on what we call it. Okay, so if we're calling it a messenger RNA, right? We call it a messenger RNA. That is the intermediary between DNA and protein. Okay, short, unstable, doesn't last long, degrades quickly. Okay, our saliva degrades them quickly. Also, RNases. Our RNA is also a thing that is what makes the ribosomal subunit. We talked about those, the ADS and 60S ribosomal subunits talked about, and the differences between the prokaryotes and the eukaryotes. So remember that our ribosomes are made up of RNA as well. And then we have the tRNAs we just talked about, transfer RNAs, transfer an amino acid to the coding chain. Right? And so those are the tRNAs as they hold an amino acid and grow the peptide chain. And with that, that concludes our structure and function of RNA video.